So, quick announcement. The exam is graded. It will be available sometime after 1 o'clock. It won't be available right at 1. The office staff will have it available in the Biochem office, ALS 2011. ALS 2011, sometime after 1. It will only be available in the office today. That is from 1 until 4, okay? If you don't get it today, that's fine, but then you'll have to get it from your TA at your next recitation. So the TAs will bring it to the recitation if you don't get it this afternoon, okay? Yes? You will need to have your ID to pick it up, yes. Okay? Yes? I can't hear you, I'm sorry. Refutation? Well, okay. If you think your exam was graded incorrectly, that's not a refutation, but if you think your exam was graded incorrectly, you need to get that in within 3 days, okay? And that starts today, all right? Now, I caution you with that people come and they think, oh, I should just get points. It's a way to get points. That's what I hear people saying. That is not a way to get points. This is done if your exam is graded incorrectly. It's not a refutation because we're not going to argue with you about whether your answer is right or wrong. All right? If there was a mistake that was made, we will regrade your exam. Understand? Okay, so that's how that works. Now, I was very disappointed in the exam. Very disappointed, okay? This was the third lowest average I've ever had on an exam. Yeah. And I honestly don't know why. I look at the exam. It does not look to me like it was any more challenging than a typical exam that I give. So I'm curious to hear your perspective. Yes? So there were some questions that you had that you didn't get answered in the recitation regarding the exam? Okay. I haven't talked to the TAs about that, so I don't know yet at this point. Which question? The pKa. The protein charge one? Okay. The proton is on the cysteine. That's correct. Yeah, that was corrected. That was changed. Yeah. So that wasn't a problem. Okay. Other comments? The average on the exam was 56. The low score on the exam was 8. There were at least 4 people who scored below 10. Okay? Yeah. The highest was 100. Okay? It was possible to do well on this exam. This was not an impossible exam. There were people who did well on this exam. So I'm a little puzzled as to what's going on. Yeah?
So does that mean like the curve is sick?
Well, I guess it depends on your notion of what sick is. But from my perspective, it is kind of sick because I don't like to see performance that low. I haven't had a chance to draw up the curve. I will draw the curve up and probably have that available by about Friday. But I was quite shocked, I have to say. And I don't see anything structural. That is sometimes I had 1 year, the lowest I ever had was in fact, there's a song about it. I had a class 1 year. There is. Biochemistry Pi. If you sing Biochemistry Pi, you'll see that it talks about a class that made 47. And they made a 47 because there was actually something that was accidental when I wrote a pKa problem for that exam. I inadvertently made it, didn't mean to, but I inadvertently made it almost impossible to solve. which is one of the reasons I say don't waste your time looking at old exams, because occasionally they have those things on them, right? And then you waste a lot of time working on something that probably you're not going to see. And so it was almost impossible to solve. I think 1 or 2 people in the entire class solved that problem. It's worth 20 points. So everybody started off out with 20 points off. And it was just a structural problem, meaning it was an accidentally written question. I didn't see that on this exam. So I'm a little puzzled as to what the difficulties were. Really am. Anyway, so I do appreciate your feedback. If you have thoughts or perspectives or whatever, there are no perfect exams. I will tell you that. I'm the first one to tell you that. I really do work to make exams be fair representations of what what I've talked about and what I think that you should understand. I do work very hard to do that. But even with that, I can tell you that there are no perfect exams. So, a low score is part my fault, it's part your fault. Okay? It'd be very easy for me to say you're a bunch of stupid people, all right? And I know you're not a bunch of stupid people, all right? So I am always curious to know how that plays out. All right. There is one thing that I can do something about, and I will. As we were going through and tallying up the grades, there were at least 20 people in here who we couldn't hardly read the scrawl they wrote for their name. Okay? Now, you guys are not 6-year-olds. Everybody should be very, very, very clearly writing their name. Okay? And very, very, very clearly writing their student ID number on there. Okay? And I'm very, very serious about this. We spent a lot of time trying to figure out who in the hay some of you were. This is not most of you. But anybody who is scrawling their name on a paper like a 6-year-old, okay, is going to pay the price. Not on this exam, but I will tell you on the next exam, if we see that again from anybody, you're going to lose about 15 to 20 points. Yeah, okay? You guys are professionals, all right? Now, I've never had to do that before. Every now and then we get a person that we have a hard time writing. But I was appalled looking at this. I'm thinking, these are adults writing their names like this. You should be proud. Maybe you weren't happy with the exam, but would you rather have a 0? Where we can't read your name? No. Okay? So make sure we are reading your name. Nice big block letters. If you don't know how to write block letters, come see me. I'll show you how to write block letters. Okay? Very serious about that. Okay? So we shouldn't be wasting our time trying to figure out who in the heck you are. Right? Okay, enough ranting. Now that's one thing that really shouldn't be done. Shouldn't be done. Yeah? Okay. Okay. So some of the stuff was fairly new, and you didn't feel like you had enough time to digest it and go through that. Okay. Okay. Yeah.
Do you have any data yet? About which questions were troublesome on the exam?
I'm still analyzing that, to be honest with you, but I also have decided to give you a chance to redeem yourselves. You don't know what I have in mind. The chance you're going to have is I'm going to ask you on the second exam. Some of the things, yes. Okay, yes. Okay? So, and I will tell you that my question that I bring, or questions that I bring to the second exam from the first exam will be based on what you did the worst on. That means if you didn't do well on some section, that's what you better hit, okay? And I don't do that to punish you. I do that to encourage you. So if you don't see things or you don't understand things, that's what I'm here for. That's what I have TAs for. Just one second, okay? And I also have something to help, all right? We are starting a brand new thing. I'm announcing it right here. We are starting a brand new thing in biochemistry that's like the help sessions that are offered for chemistry, math, physics, and biology. We're starting one for biochemistry. It's called the Major Groove. Cute name, right? Okay. And the Major Groove will have some biochem students who will staff tables and come and be available for you to come and ask questions. This will be in the evenings. Okay. I don't have the location set up quite yet. I'll be announcing that in the next day or two. And that's completely free. You come, you get the help that you need from them, et cetera, et cetera. And that'll be held probably about 10 to 12 hours a week. Okay? So there's a fair amount of help that you can get. The TAs are available for free. I've had a couple of you ask for tutors. And I'm also happy, I've got an excellent tutor if you want to pay, if you want to pay someone to help you to learn the subject. I've got an absolutely excellent tutor I can recommend to you as well. And that would be one-on-one that you would get with her. Okay? So again, I don't do this to be, any of these things to be mean. I really want you to learn this subject. It's really important that you do that. And so encouraging you to master this material is important. So I want to see you do well. So you will see something, 1 or 2 questions on the next exam from the first exam. Okay. Now the euphoria is gone. Okay, well, perhaps to bring it back, I'll bring a limerick. As the photon took off for his flight, he packed up his things really tight. When asked why he dragged just one duffel bag, he said he was traveling light. Okay. Hopefully I put everybody back in a good mood. Now, last time I went through a fairly detailed description of the mechanism of action of chymotrypsin, and that mechanism of action of chymotrypsin is almost identical to the mechanism of action of any of the serine proteases. In fact, for our purposes, you may consider them identical for all the serine proteases. Okay? There are related proteases that you will see actually use similar mechanisms to the serine proteases. One is known as the cysteine proteases. If you think about it, serine, which is what gives the serine proteases its name, has an OH group at its active site that plays a critical role in the catalytic cycle. Cysteine, in a cysteine protease, has a SH, a sulfhydryl, in the active site that plays a role. And I'm going to show you a mechanism for that in just a minute. All right? That is equivalent to the OH that we see in the serine proteases. The cysteine proteases don't have a catalytic triad. They only have 2 of the 3 amino acids. They have cysteine and histidine present in the active site of a cysteine protease. And again, I'll show you that in a second. A second group of proteases that at first glance may not look like the serine proteases are the aspartyl-proteases. And these are proteases that have 2 different aspartic acid residues in the active site of the enzyme. Okay? And we'll see that they use an activated hydroxyl group that's a nucleophile that's very much like the hydroxy, I can't even think, the alkoxide ion that we see in the serine proteases. And the last of these that I'll mention is something called the metalloproteases, and these have a zinc that plays an important role in activating the protease, as we shall see. Well, the strategies by which these work are shown here, and they're, as you will hopefully begin to see, very, very similar. So cysteine proteases, as I said, have a sulfhydryl side chain that gets activated during the mechanism of action of this set of proteases, and it works in the same way, essentially the same way. When the protease binds to the right substrate, a slight shape change occurs. When that slight shape change occurs, the proton of the sulfhydryl is brought into close proximity of the histidine, which pulls it off, leaves behind an activated sulfur, which is a nucleophile that attacks the carbonyl just exactly like you saw in the serine proteases. So with the exception of missing an aspartic acid in the active site and having a sulfhydryl instead of a hydroxide, the mechanism is essentially identical to that of a serine protease. Some of the players have changed, but the mechanism doesn't really significantly change. If we look at the aspartyl-proteases, they're kind of interesting by comparison. Okay? And I want to talk about the mechanism of these for a minute and then make you think a little bit about how they work and how their mechanism of action is similar but also different from that of the serine proteases? Well, at first glance, they look quite different. Okay? We don't have a hydroxyl that is there as an activated intermediate. We instead have the side chain, or two side chains of aspartic acid. Okay? Two side chains of aspartic acid. Here's one, here's the other. This one is shown with a proton on one of the groups. You don't have to worry about that for our purposes. That's not really critical. And it doesn't really matter which one of these that we're talking about, because from the appearance of what's on the screen, it's arbitrary anyway. But the important point is that the binding of the proper substrate at the active site causes a conformational change within the protein that brings this negatively charged carboxyl group into close proximity of a water molecule that's in the active site. Okay? Into close proximity of a water molecule that's in the active site. And as you have seen before, that proton of the water molecule is extracted from the water, leaving behind a hydroxyl that is negatively charged, that is very reactive, just like the alkoxide ion was very reactive. And that hydroxyl attacks the carbonyl carbon of the peptide that's in the active site, just like you saw before. So it's a nucleophilic attack, just like before, only this is now a hydroxyl instead of an alkoxide ion, but the result is the same. The attack of that carbonyl carbon causes the peptide bond to fall apart, just like it did in the serine protease, and then the peptide is released. Now, there is something about this mechanism, even though it's using this similar mechanism to the alkoxide ion, there's something about this mechanism that is different about the way that it works. My question to you is, first of all, do you recognize what would be different based on what I've shown you here compared to what you saw in a serine protease? Yes, Garrett. Is it related to the concentration of water? No, it's not. Yes? No covalent binding. Excellent. That's exactly it. All right? When we had the alkoxide ion, the other half of that oxygen was linked to a carbon because it was the side chain of a serine. That negatively charged oxygen that was attached became covalently linked to one of the peptides, right? So we saw a fast step, which was the breaking of the bond and the release of in the case of the yellow color. But then we had a slow step where that bond between the side chain of the serine and the peptide had to be broken. This guy will not have a slow step, right? Because when this attacks the carbonyl group, okay, the bond is broken. There's nothing left to attach to on the enzyme. And that's what we saw in the serine protease. So this guy will just go zipping right through its way and won't have that slow step like you saw in the serine protease. So very good. Comments, questions? Okay. The last group of the proteases I want to talk about here are the metalloproteases. And the metalloproteases are, again, using a sort of a similar mechanism. They use, usually, at their core, an atom of zinc. Okay? Zinc is an essential nutrient. Zinc is used by a variety of enzymes. And here is one use of zinc. The zinc is coordinated, or essentially, if you want to think about it as linked, to some histidines that hold it in place. We're in the active site. Here's the polypeptide chain. And here's a water molecule that's made its way into the active site. Okay? Over here, there's something labeled B. And B is their clever way of putting the word base. All right? I don't like using that word, so we'll call B bonehead. Okay? Bonehead is on the side. All that bonehead does is it has a relatively negative charge. That's indicated by the presence of these electrons that are out there talking to the protons saying, hey, look at me, look at me. It's called B because depending on the enzyme, it could be a variety of different things. So it's some electron-rich component of the enzyme in the active site that interacts with one of the protons of the water. And notice the water is being held in place by the zinc. That's the only function that the zinc has here, is holding the water in place. Okay? The B pulls the proton off of the water, and that creates, there's our old friend, that reactive hydroxyl group. That reactive hydroxyl group is a nucleophile. It attacks the carbonyl carbon, and everything else that we've talked about before all happens again. So when you learned one mechanism for the serine proteases, you learned a general mechanism by which each of these enzymes borrows from. Each of these enzymes uses some component of that mechanism that we saw in the serine protease. Okay, questions about that? Yeah? Yep. That's the rest of the polypeptide chain there. Yeah. Yes, Justin.
So the primary common thread between all of these is the carboxyanion and how that leads that.
Correct. So his comment is that the common thread in all of these is there's a carboxyanion of some sort, and that carboxyanion, I'll take it further, is a nucleophile that attacks a carbonyl carbon in the polypeptide chain. That's common to every mechanism. Yes? So the oxyanion hole was something we saw in the serine protease. Actually, let me back up and clarify something I just said to Justin, which isn't quite right. They all use carboxyanions. Well, they use, I don't want to call it carboxyanions, no, because this is not a carboxyanion, obviously, and a sulfhydryl chain is not either. So they're using anionic substances that are nucleophiles. Let's use that common term, okay? Now, the oxyanion hole is something that's a part of the serine proteases that stabilizes the unstable intermediate that arises by the nucleophilic attack of the alkoxide ion in a serine protease. You could imagine similar type structures in all of these proteases that will help stabilize that unstable molecule so that it falls apart in the desirable way, which is to break the peptide bond. And that's what the function of the oxyanion hole is to keep that reactive intermediate from reacting in a way that would be undesirable for the enzyme. Make sense? So we don't want to make a suicide inhibitor or a suicide reaction. That's to keep that from happening, essentially. Other questions? Yes, up above. Does this what? Does this form a covalent bond? You betcha, because the hydroxyl is attacking right there and becomes covalently attached in the process. You bet. Okay, so those are the different proteases that we talk about here. I want to spend a little bit of time talking about an interesting enzyme. We've talked around it a little bit, and that's carbonic anhydrase. Carbonic anhydrase is the enzyme that when I showed you the different KCats, had a KCat that was up to about 1 million molecules of product per molecule of enzyme per second. An almost unfathomable rate. Okay? Understanding the mechanism of that enzyme is therefore kind of interesting. And you'll notice that when I showed it to you before, it showed a couple of different KCats. KCat is usually a relatively constant value for an enzyme. And carbonic anhydrase doesn't have it actually has a fairly variable KCat. Let me remind you first of all what it does, and then I'll tell you about the enzyme. Carbon dioxide plus water. Carbon dioxide, of course, is a byproduct of metabolism in your cells. It's a toxic substance. Carbonic anhydrase combines with water to convert that into carbonic acid. And then carbonic acid is fairly acidic. It loses a proton quite readily and becomes bicarbonate plus a proton. So this happens in your blood. It's one of the things that actually helps to stabilize the pH in your blood through a rather complicated mechanism I won't go into. But suffice it to say that this enzyme is pretty critical in making this happen, and also, in a sense, detoxifying the carbon dioxide that's there. So not all the carbon dioxide gets carried by hemoglobin. We saw some of it being carried by hemoglobin. The rest of it gets made into bicarbonate, as you see right here. Now, this enzyme is interesting. Okay? This shows how the KCat of the enzyme varies as a function of pH. Why should it vary as a function of pH? Shouldn't the KCat be something that's reasonably constant? Because we talked about it being a constant for an enzyme. All right? Well, it turns out that if we look at the pH, measure the KCat of this enzyme at a pH of about 6, we see that it's maybe got a KCat of 50,000, meaning 50,000 molecules of product per molecule of enzyme per second, which isn't bad, okay? But if you take this puppy all the way up to 9, you can see a pH of 9, you can see that its KCat jumps up to 1 million. 1 million molecules of product per molecule of enzyme per second. Now, why is there this very wide change of KCat? And it turns out that the enzyme, all right, is assisted by a higher pH in its mechanism. Okay? Here's what happens. We're looking in the active site of carbonic anhydrase. And lo and behold, what do we see there but this zinc, like we saw in the metalloproteases. Okay? The zinc is held in place by 3 histidines, kind of like we saw in the metalloproteases. And that zinc is helping to hold onto a water molecule, kind of like we saw in the metalloproteases. Okay? The first step in this process involves loss of a proton from water, and that's an ionization. Water, you should recall, ionizes at a relatively low rate, about 10 to the minus 7th moles per liter of water, which is why pH of 7 of pure water exists. Okay? If we increase the pH of water, what happens to the likelihood of water losing a proton? It goes up. The higher the pH, the more likely water is going to ionize, and consequently, the more likely this first step of this reaction is going to occur. The reason that the KCat changes so dramatically is because this very first step is facilitated by a higher pH. Okay? Well, we have a hydroxide ion, a reactive hydroxide. It's a negatively charged hydroxide at that point. And you know from what I showed you in the metalloproteases that it is a nucleophile. And what will it attack? It will attack a carbon dioxide, also brought into the active site of the enzyme, to in the reaction, as you see here, to create this carbonic acid. Okay? So, ionization, attack, linkage, release, and you're done. If this step, step number 1, is not occurring very rapidly or not occurring very extensively, then the enzyme is slower, because the enzyme has to have a way to make this thing pull off. You've seen one already. We had the B in the metalloproteases. Okay? But if we increase the pH, we help the enzyme out a lot in terms of making this actually occur. Okay. So, pretty cool process, pretty cool reaction. Question about that? Yeah? What causes the carbonic acid to leave right here? It's actually released and replaced by water. So it's the equivalent of a hydrolysis that's coming back in to release that guy and recreate the original structure. Don't know off the top of my head. Yeah. Yes? If this is used to control pH in the bloodstream, is this why it only happens at high pH? It happens at lower pH, as you saw. Okay? So the control of pH in the bloodstream is actually a fairly complicated process, but the formation of carbonic acid does play a role in that process. It's not this reaction that's causing the pH to be relatively constant. It's actually the action of carbonic acid as a buffer that's helping that to happen. Okay? Yes, Deidre? Okay? Deidre's asking the next question I was going to ask you. So my question was, if we looked at right here, what do you expect would happen if we continue to increase the pH? What's that? It'll plateau and it'll come down. Why will it come down? Come on, folks. Make me proud of you. What's that? Denaturing. Yeah, of course. You get to a high enough pH, any big change in pH away from physiological pH is generally going to denature enzymes because you're changing the ionic environment, and you change the folding, you change the shape, just like you do if you add a detergent or something. It's going to denature, and you're going to see this guy come back down. So this doesn't go up forever. Yeah. Okay. That's carbonic anhydrase. Excuse me. Let's see. The next thing I want to talk about are restriction enzymes. And restriction enzymes are very, very cool. Okay? Restriction enzymes are enzymes that recognize and bind to and cut DNA at specific sequences. Recognize, bind to, and cut DNA at specific sequences. We remember that DNA, of course, has 4 bases. And just like we said that the sequence of amino acids for a protein was critical to the protein's function, so too is the sequence of bases within a DNA molecule critical because it's the DNA that's carrying the genetic code. Okay? Let's imagine that I am a virus. And I'm a special virus. I'm a bacteriophage. A bacteriophage is a virus that infects bacteria. We commonly just call them phage, P-H-A-G-E. All right? A phage infects a bacterium, and when it infects a bacterium, what it does is it attaches to the bacterial cell, just like viruses do with our cell, injects its nucleic acid into the bacterium, and the virus's DNA contains coding for various viral proteins to allow the virus to propagate, just like it happens in our cells. Okay? A bacterial cell that gets attached to by a virus is in pretty deep doo-doo. Okay? Because here's this viral DNA coming in doing its thing, and our cells have a similar problem, but with us, we have a protective mechanism. We have a protective mechanism that's known as our immune system. We may lose a few cells, and we do. We catch a cold or whatever. A virus is killing a bunch of our cells. But our immune system is ultimately recognizing that virus, attacking that virus, and protecting the rest of the body so that the rest of the body doesn't get completely taken over by that virus. Okay? This is even true for Ebola, by the way. Okay? The main issue with Ebola is not your immune system. The main issue with Ebola is keeping the person alive long enough so that the immune system can protect. Okay? There's been actually several interesting articles about that, keeping the patient alive long enough so that the immune system can catch up. And that's why not everybody dies from Ebola. It really depends on how well you can keep people alive in that process. There's only been one person in the U.S. who's died from Ebola, and there have been, I think it's 6 or 8 people now that have been infected by Ebola that are U.S. or of U.S. descent. And on average, Ebola kills half or more of the people who get it, right? So people who've been treated here by this they've been able to keep them alive because they have more support and other ways of keeping people alive so that Ebola is not necessarily a death sentence. Okay, so anyway, that's another matter. We are protected by our immune system. Well, a bacterial cell, okay, although we're learning that they have some remnants or something like an immune system, a bacterial cell in the simplest scheme doesn't really have an immune system. That virus goes in, and that cell's hosed because there aren't other cells around it that will protect it. It's not a multicellular organism. Well, bacteria are the ultimate lone wolves. Okay? They've got to do everything themselves. And one of their means of protection are restriction enzymes. Restriction enzymes are enzymes that are made by bacteria. And as I said, they recognize, bind to, and cut DNA at specific sequences. They actually protect bacteria. That's why they exist. They're part of what we call a restriction/modification system. They're part of a restriction/modification system. I'm a virus. I inject my DNA in the bacterial cell. My DNA has sequences in it, and if the restriction enzyme that's in that bacterium recognizes those sequences, it will bind and cut that viral DNA. Cutting the DNA of the virus essentially inactivates it. Protection. Bang. Very simple, very good, as long as the bacteriums, the bacteriums, it's a possessive, as long as the bacterium's restriction enzyme recognizes a sequence in the viral DNA, everything's fine. The restriction enzymes typically recognize relatively short sequences of DNA so that on random, you will see many of those sequences within a viral genome. Bingo, you got it. Okay? Well, then one question arises, how come it doesn't cut up its own DNA? Right? Because if it's there to cut sequences, and that sequence occurs several times in a viral DNA, which is much smaller than the cellular DNA, how come it doesn't cut itself up? And the answer to that question has to do with the modification part of the system. Okay? It has to do with the modification part. Bacterial systems have restriction enzymes, that's one half of that system, and they also have a set of enzymes called methylases that are the other part of the system. That's the modification part. So a restriction modification system consists of a restriction enzyme and an associated methylase. All right? Now, I haven't shown you a figure yet, and the reason I haven't is we don't have any good figures to show you the overall thing. But I'm going to show you. That's not the one I want. Okay. I'm going to show you one of the enzymes, one restriction enzyme. Okay? There are hundreds of enzymes, each of which recognize, bind to, and cut specific DNA sequence. This is one called EcoR5. It's found in some forms of E. coli. And it recognizes the sequence GAT-ATC. When it sees that sequence, if there's nothing else on that sequence, it will bind to it and it will cut it, and that's that. It doesn't matter if it's a cellular DNA or if it's a viral DNA. So how does the cell get protected? By the methylase. Cellular methylase, the methylase that corresponds to EcoR5, for example, also recognizes the same sequence that the restriction enzyme recognizes. So the methylase will recognize a GAT-ATC just like the restriction enzyme will. But instead of cutting it, what it will do is it will put methyl groups on that sequence. Methyl. It puts methyl groups on that sequence, and when it does that, the restriction enzyme can no longer recognize, bind to, and cut that sequence. This is a select system, guys. Now, one of the things students always get puzzled about is they want it to be perfect. It's not perfect, as we will see. But it's very, very good. Just a second, okay? The cell is sitting here with all that DNA, and it's got both enzymes present in it. As long as the methylase recognizes and binds that sequence first, the cellular DNA is fine. It's going to be protected. Okay? If the restriction enzyme finds it first, then that's going to get cut, and the cell's going to be gone. Well, there's a lot of bacteria. Obviously, the methylase is doing its job pretty well. The bacteria aren't committing suicide too frequently. And the bacteria are protected. The flip side of this is when the viral DNA comes in, if the methylase finds it first, what's going to happen? protection. Okay? It's not a perfect system, but it's a very good system. If you've ever worked with DNA in a laboratory with bacteria that have a restriction modification system, what you will see is that if you try to put DNA into cells, and there's techniques that will do that, if you try to put DNA into a cell that has a restriction modification system, it's hard. And that's because that the restriction part of the system gets to it and cuts it very quickly. I cut you off earlier. What was your question? It could. It could. So it's not, as I said, it's not a perfect system. So it could. Yes? Are methylases and restriction enzymes floating around the cell? The answer is yes, they are. Yep. Yeah, back there. Yeah, good question. So if you wanted to increase the efficiency of getting the DNA into the cell, could you methylate it beforehand? That's actually one thing that people do sometimes. Yep. And the other that they do is they will just do a lot of cells. And what you see is you have a very low efficiency, but once you've got one, then it's got the methylase and everything is fine. So, yeah. She said, we'll do the same thing to a plasmid. I'm talking about plasmids. So it's, when we put DNA into cell, it's typically plasmids that we're putting in. Yes? Well, that's a very good question. Okay. So the question is, wouldn't it be better for the cell to have the methylase around where replication is occurring than just floating around the cell? I would agree. And it might. We don't fully know the nature of the replication fork that you'll hear about when we talk about replication of DNA. There are some suggestions that there are some complexes of proteins around the replication fork that may help facilitate efficient replication. This is one you might imagine in that sense, okay? Although I've never really thought about it, to be honest. Others are the things that make nucleotides. In the things that make nucleotides, it looks like they feed the replication fork, and they're located very close to the replication fork. So that's another consideration of it. Very good question. Okay. Well, I want to just briefly talk about the mechanism that these guys use before abandoning that. So what's happening in the cutting of DNA by a restriction endonuclease is that a phosphodiester bond is being broken. Okay? A phosphodiester bond is breaking the linkage in the backbone of a strand. So there's a deoxyribose, there is a phosphate, and there's another deoxyribose, and each of those is attached to a base. So we're looking at one strand. Restriction enzymes cut double strands, and what they do is they cut once on this strand, and they cut once on the other strand, and that's how the break is introduced into the DNA. Okay? The break has happened here. It's a hydrolysis reaction. And a hydrolysis reaction is interesting. The hydrolysis reaction uses an activated hydroxyl from water. Okay? You've seen this activated hydroxyl popping up several times now. Restriction enzyme is using that. So part of the mechanism of action of a restriction endonuclease is it must activate water. Make it a hydroxyl that is reactive. It's a nucleophile. Okay? Now, this shows the double-stranded DNA, and it shows, at least schematically, the binding of the restriction enzyme to the DNA. Now, the figure, I don't like the figure very well, so I'm just going to describe to you what happens. Okay? Restriction enzymes, the way they work is they've got to find the sequence that they cut. And they don't just randomly go and bump into a DNA and find the sequence. In fact, it's much more efficient for them, once they bump into DNA, to grab a hold of it and start sliding down the DNA. They actually slide down it, and as they're sliding down it, they can technically read the sequence that they're binding to. So by going at one end and then sliding down until they find the sequence that they cut at, they just go zipping along. And all of a sudden, they hit the sequence that they cut at, and boing, stop. Now, what you've seen of all the enzymes so far is what happens when the proper substrate gets into the active site. What happens to the enzyme? Slight change in shape, right? The same thing happens in the restriction endonuclease. The restriction endonuclease has found and bound to its proper site. A slight change in shape occurs, and when that occurs, the DNA is actually bent by the restriction endonuclease. There's a bend that is put in place, and that bend turns out to be essential for the activation of water. Because the bend allows an ion of magnesium held by the enzyme to hold on to a water so it can get activated and break the phosphodiester bond. That bend is essential. Okay? How does the modification system protect it? It protects it by partly preventing the enzyme from recognizing the site. The enzyme doesn't recognize the site, the bend cannot occur, and meanwhile it just goes sliding along as if it didn't exist. Make sense? Yes, question? How does this interact with transcription? Typically, when you're doing this in a laboratory, of course, you're doing it apart from the process of transcription. But this would have essentially little or no effect on transcription that was occurring in the cell. Would the RNA polymerase be interacting and interfering with this? It could to a slight extent, but probably not anything significant. Okay, it's time for a song to finish. You guys, I'm singing a song here for the first time ever. It's the second time in about 3 days I've got a brand new song. This one, anybody know the song Chim Chim Cheree? Okay. I'm obsessed with AAGCTT because it's the binding site of Hindi3. That's the restriction endonuclease, by the way. Cutting up DNA most readily. The ends are not blunt when they're cut up, you see. 5' overhangs of AGCT. Bacteria don't have an immune system, so they must fight off phages or they will not grow. Protection by chopping is their strategy, and one of the cutters we call Hindi3. On binding to AAGCTT, the site recognition site's bent easily. Phosphodiesters attacking, meanwhile, has water behaving as nucleophile to stave off the phage for a little while. Why don't these enzymes cut cell DNAs? The answer's provided by amethylase. Adding a methyl group on top of what the sequence these enzymes would otherwise cut. So cells get protected in this simple way from nuclease chewing of their DNA. Phage is not lucky in most every case unless methylases win the enzyme race. If that happens, then the cell gets erased. All right.